Welcome to the Chemistry, Biology and Math Revision Hub. Today we are discussing the Pearson Edexcel International A Level Biology Unit 4 for January 2021. This video is part 1 of this paper, so in the next video we'll discuss part 2. And in part 1, this video, we are discussing question 1 to question 4. In the next video, we'll discuss question 5 to question 8. Do not forget, I will put the link in the description box of this video to part 2, so please do not forget to check it out. Let's begin. Question 1 says, inflammation is involved in the non-specific response of the body to infection and injury. Complete the diagram to show the 4 characteristics of inflammation. When there is inflammation, there is going to be swelling. Sometimes you're going to feel pain or aches or throbbing that could be felt. This is to alert you that you've hurt yourself. Then there is going to be redness of skin. Redness of skin is there because more blood has been diverted to that area that is inflamed. And then there is increase in temperature. This is so that enough energy is provided for the reactions that occur during inflammation to occur appropriately or to occur at the right amount of energy or right temperature. The next part says, inflammation occurs following a cut to the skin. Describe the role of 2 of these characteristics of inflammation in response to a cut skin. So one thing we know is, since I've told you, I've written the 3 characteristics here. So the pain is felt in order to alert the person of injury or that the injury has occurred. The pain is there to alert so that you take care of to prevent infection basically. And then the increase in temperature or increase in hotness, it is there to provide the right amount of energy so that the rate of reaction is going to be sped up. Remember, phagocytosis uses enzymes that are going to break down in case any pathogens or any things causing infection. So if this is to occur, the enzymes have to be activated enough in order for reactions to occur. Another thing is cell division. Cell division, skin cells that have to be made to regenerate the area that has been hurt have to also divide by mitosis, and this requires a lot of energy. So the high temperature is to allow that kind of reaction to occur. The next part is swelling. Swelling is going to be because more blood is diverted towards the area that has been wounded in order to supply you with blood cells and increase the cells required for phagocytosis and so on. Redness is as a result of more blood-containing blood cells, antibodies arriving at the site of the injury. So all these are working together to ensure that the area that has been hurt is healing faster, and so that infection is going to be prevented. Let's go to the next part. Part B, anti-inflammatory drugs reduce inflammation. Turmeric is a spice that is added to food. Curcumin is a chemical in turmeric that has been shown to reduce inflammation. The graph shows the effect of treating inflammation with curcumin extract and an anti-inflammatory drug A. So here we can see the data in this graph. We see the y-axis is showing inflammation, while on the horizontal axis we have days after treatment. So this is day 1 until day 6 for drug A and for the curcumin extract. So overall, we can see that the curcumin extract has a greater chance or greater ability to bring down the inflammation in comparison to drug A. As you look at this data, generally you can see because after day 6, it had the greatest decrease in comparison to drug A. So the first question says, compare and contrast the effect of treating inflammation with curcumin extract and with drug A. When they ask you to compare and contrast, they want you to talk about the similarities as well as the differences. So I began with a similarity. Overall, both decrease inflammation with time. There is overall, they're going to decrease with time. This one doesn't matter even if there is a slight increase here. Usually when they ask you to talk about similarities and differences, you can look at the bigger picture. The bigger picture, the beginning and the end, we can see there is a general decrease for both substances used in this treatment. Then the next part, I say, however, curcumin extract is more effective at reducing inflammation than drug A. If we can compare, by day 6, you can see the inflammation is way down in comparison to the way it would have been. Imagine if this was day 1, it means if before day 1, it could have been as high as that. So the percentage reduction caused by curcumin extract is greater in comparison to the percentage reduction brought about by drug A. And that brings into my third point, the maximum achieved decrease. This is kind of the percentage decrease. in inflammation is greater in curcumin than in drug A. And 2 days after treatment, can you see here? This is what I'm talking about. 2 days after treatment, we can see there is a slight increase in the inflammation when you use drug A, while the curcumin extract causes a decrease as well. Although here too, there is a slight increase. So you can talk about this data or you talk about that data, you will get the same marks. So I say 2 days after treatment, the inflammation increases for drug A, but curcumin extract shows a decrease in inflammation. So I hope this makes sense. I will go to the next part. So they say many treatments use 1 gram curcumin extract, and they say a sample of turmeric contains 3% curcumin. So if they use 1 gram and this is 3%, it means if it was a mass ratio, it's going to be 3 grams of curcumin in 100 grams of turmeric. I hope you can see this. This will be curcumin. It means if you need 1 gram to treat somebody, you're going to have to divide 100 by 3, creating a 33.3. So here they said which mass of turmeric, which is the mass of turmeric for 1 person. 1 person, we will need 1/3 of 100. If 3 grams of curcumin are contained in 100 grams of turmeric, 1 gram is going to be contained in 100 divided by 3, which is 3.3. And the answer here is going to be a C. So this brings us to the end of question 1. Let's move on to question 2. Question 2. Antibiotics are medicines used to treat some medical conditions. The table shows some medical conditions and whether or not antibiotics are needed to treat the condition. So we see the medical condition, we see impetigo, yes. Whooping cough, yes. Middle ear infection, sometimes. Sinus infection, sometimes. Multiple sclerosis, no. And rheumatoid arthritis, no. So we're going to look at the questions they're asking. So the first one says, explain the use of antibiotics to treat these conditions. So it means we have to talk about one condition at a time. Oh, basically we can pair them that, and that. So I said for impetigo and whooping cough, antibiotics are used, yes. So because there is a confirmation that yes, they always use antibiotics, it means these 2 conditions are caused by bacteria. So impetigo and whooping cough are treated with antibiotics because they are caused by bacteria. Now these are sometimes treated with antibiotics because they are sometimes caused by bacteria, not always. Some other times it could be something else. So I say middle ear infection and sinus infection are sometimes treated with antibiotics because they are not always caused by bacteria. If they are caused by other pathogens like viruses, then the antibiotics will not work. It means that time something else has to be used. Then lastly, we have multiple sclerosis and rheumatoid arthritis. I say these are never treated with antibiotics because they are never caused by bacteria. So that's it. Never caused by bacteria, so you cannot use antibiotics. The key thing here is antibiotics are there for treatment of infections caused by bacteria. I will go to the next part. This part here says, the graph shows the effect of 3 drugs. There is drug J, K, and L on the number of bacteria in a culture. So we have the graph here. There is the number of bacteria on the y-axis, and we have time on the x-axis. So here we can see, if you can see the y-axis is basically number of bacteria. So here the number of bacteria will increase. If this is a medicine causing the number of bacteria to increase, then that's not an antibiotic. If there is a medicine causing the number of bacteria to decrease, that is an antibiotic, and it should be bactericidal because it kills them. Now there is another drug that maintains. There is an increase, but the increase is not as great. So this is going to be an antibiotic, or at least it has antibiotic effect. And I call this a bacteriostatic because it manages the growth of the bacteria. This one here kills or destroys the bacteria completely, or decreases them over time. So here they ask, which row of the table describes each of the 3 drugs? As you can see, drug J is not an antibiotic. Drug K is going to be an antibiotic, but it's a bacteriostatic. And then drug L is going to be a bactericidal antibiotic. So let's continue to the next part. This part here says, one problem of taking antibiotics is their effect on gut flora. Gut flora are like microorganisms that survive symbiotically in our digestive system. So in the gut, basically. The diagram shows the effects of three antibiotics, P, Q, and R, on the proportion of different types of gut flora. Each section in each of the diagram represents the different types of gut flora. So let's see, there is type A, there is type B. Ignore these values here. I'll talk about them later. So they say, explain the role of gut flora in protecting the body from infection. Now guys, the gut flora, or the microorganisms we have in our gut, they are there to help us. Actually, they benefit, we also benefit. So it's a mutual relationship. These gut flora, they are not pathogenic. However, their role is to compete with pathogenic microorganisms or pathogenic bacteria. In the case you have maybe eaten food that is contaminated, the bacteria will not have the same space. Basically, the gut flora will try to outcompete the pathogens for space as well as nutrients to prevent them from accumulating in your gut and causing you disease. So here, the pathogenic bacteria in the gut, they compete with them for nutrients and space, and this prevents growth and increase in the number of those pathogenic bacteria. So basically, your body is being protected by the gut flora. Then here they say, explain why antibiotics can affect gut flora. Remember, these are antibiotics, and the gut flora are bacteria. Since antibiotics kill or destroy bacteria, they are not selective when they are doing that, except if specific bacteria have resistance to that specific antibiotic. So if not, then there is no selection. They will destroy everything in their path. So I said antibiotics affect bacteria. Since gut flora are bacteria, they can be killed. Also, antibiotics are not selective when they destroy bacteria. Only those bacteria with resistance to specific antibiotics will grow and increase in number. So those that do not have resistance will be completely destroyed. Let's continue to the next part. The number of type A in gut flora in the absence of antibiotics is 6 million. Estimate the number of type B in gut flora in the absence of antibiotics. And they say give your answer in standard form. For both, it's in absence of antibiotics. So I will go back to my pie chart so that we can try to see which percentages. So here, this is with no antibiotics, and they are looking at type A and type B. Again, I used the protractor using the protractor on the iPad. So this is an estimation. However, also in the exam, of course, there is some estimation. So this is like 63 degrees, and this is like 107 degrees if the total is going to be 360. So I used for type A 63, and that is 107. That is the angle I measured. So let's go back to this part. And then here I say for 63, the proportion of type A is going to be 63 over 360 times this is going to be times the number. This is the total number of bacteria and the total number of gut flora. And again, here you see I said where X is the total number of gut flora. So from the total number of gut flora, type A is 63 out of the 360. So this gives me 63 over 360 times X should give me 6 million, and then make X the subject, giving me this value here. And then, so this is the total number of all the gut flora present. So since I know the other degree, 107 for type B, 107 over 360 times the total should give me the number of type B gut flora present, so which is 1.01 times 10 power 7, because they say the answer should be in standard form, so I wrote in standard form, and that was my answer. So lastly here they say, deduce the effects of antibiotics P, Q, and R on gut flora. If you look at the different pie charts that were given, you can observe that antibiotic P kills all the other types except 3, and then only 3 survived. All the other types were destroyed. For antibiotic Q, it killed 2 types of the flora, and then antibiotic R had no effect on the gut flora. So that is what you can observe from the pie charts. So this brings us to the end of question 2. Let's go on to question 3. Question 3. In 2018, an organization carried out a global survey into the attitudes of people towards vaccines. Vaccines provide artificial active immunity. Describe how a vaccine results in active immunity. Now we know vaccines contain attenuated or weakened versions of a pathogen. So if this version enters the body, macrophages are going to engulf it. And when they do engulf it, they will sort out the antigens and present those antigens on their surfaces. So when the antigen-presenting macrophage comes into contact with a specific T cell, this T cell could be activated to become a helper T cell, and then the activated helper T cell, of course moving through the body, could come into contact with an antigen-presenting B cell, and this helper T cell will release cytokines, and these cytokines will cause the activation of the B cell, and that B cell will actively divide by mitosis, forming many clones of itself, which we call the plasma cells. These plasma cells will have a lot of the rough endoplasmic reticulum present in them, so that a lot of antibodies can be produced by these. So that is active immunity, because the antibodies have been produced. However, any B cells as well as T cells that are retained in the body, meaning those that are not used in the immune response, will remain in the body as memory cells. And in the case if there is any reinfection by the same pathogen, it means the memory cells can actively respond by dividing rapidly or becoming rapidly activated and producing antibodies in large volumes within a shorter period of time, so that immune response is quick and the person does not get diseased, basically. Part B, this survey included 3 aspects: the trust that people have in scientists, their thoughts on the safety of vaccines, and their income. So they say the graph shows some of the results of this survey. So here we have income from low to high. We have the trust in scientists from low to high. We have the key here. And the first question is asking us to identify 3 conclusions that can be drawn from the results of this survey. So the first conclusion we can see is going to be overall. Overall, we can see all income brackets they agree that vaccines are safe. And again, you can see safety of vaccines is this black thing. In all income brackets, we have that black thing showing that they all agree that vaccines are safe. And then we see the next part is, I say that those that agree that vaccines are safe have the highest trust in scientists. Yes, these agree that vaccines are safe. Their trust in scientists is going to be the highest. The same thing here, same thing there, and the same thing here. Those that do not agree that vaccines are safe, of course, they have the lowest trust in scientists, you can see overall. And then the next part I said high income earners have the lowest trust in scientists, because you can see for most of their results, the high income earners, their vertical axis is the lowest. Whether for the agreeing whether vaccines are safe, they don't trust scientists. Whether those that neither agree nor disagree, they do not trust scientists as much. And those that disagree that vaccines are safe, their trust in scientists is lower in comparison to the other groups. So those are the 3 conclusions that I drew from here. I will go to the next part. The next part says, suggest 1 reason for the different attitudes of these people. Now, different attitudes could be due to different exposures or different friends, or you watch news or you read newspapers, whichever. So it depends on people or things in your life that can influence you. So it could be beliefs. So I say people's beliefs can be influenced by friends. It could be families, it could be media, it could be anything. Anything the person is exposed to can be able to alter their beliefs, and they act in a certain way towards a specific thing. So part C says, the more people in a population who are vaccinated against the disease, the less likely it is for non-vaccinated people to become infected. This suggests why vaccination is more successful when a greater proportion of people are vaccinated. Now, the key thing is, when people are vaccinated, even if they are infected, their immune system is strong enough to fight in order to prevent the disease symptoms from being exposed, or from coming out, or from the person feeling the symptoms of the disease. So if the person does not feel the symptoms of the disease, there is a high chance that they will not be active spreaders. So if they are not active spreaders, then there is a lower chance of them spreading to non-vaccinated people. So imagine if the bigger portion of the population is vaccinated, it means you have very fewer people who have the chance of catching the disease and therefore spreading it. So that is why vaccination is going to be helpful. So here I said, people who are vaccinated are less likely to develop the disease. This means the symptoms. So they could be infected, but their immune system can fight really fast in order to prevent the disease symptoms from being coming out, or for the person from experiencing the symptoms. So then I say the higher the number of vaccinated people, the fewer the infected people, and the less likely the unvaccinated people come in contact with the disease spreader. So this brings us to the end of question 3. Let's move on to question 4. Question 4. Greenhouse gases are involved in anthropogenic climate change. A1 says, which of the following are greenhouse gases? So the greenhouse gases, of course, carbon dioxide is, oxygen is not, methane is, water vapor is, carbon dioxide is, oxygen not, water vapor is, methane is, carbon dioxide is, and oxygen is not. So we can see it's B, methane, and water vapor. The next part says, state what is meant by anthropogenic climate change. Anthropogenic climate change is the change in the average temperature or weather patterns due to human activities. So these could be, the anthropogenic part of this question is human activity, and the climate change part is change in weather patterns. So the extra part I added on could be deforestation. Humans can carry out deforestation or cause deforestation that leads to anthropogenic climate change. It could be combustion of fossil fuel and many other things that could lead to the anthropogenic climate change, basically. So let's go to the next part. Here they say the distribution of species has been affected by climate change. The diagrams show how the distribution of one species in a country in the northern hemisphere could change as a result of climate change. So we have the diagram here. They say distribution of species before climate change. So before climate change, the distance from the north of the equator, or the distance north from the equator, was not that big. It was meaning they were closer to the equator. However, the distribution of the species after climate change, we can see the distance is greater from the equator. So the question is asking, explain the changes in the distribution of these species in this country. When they ask you to explain something, you have to state the fact and give a reason why that fact is the way it is. So the fact we have here is the species moved north away from the equator after climate change. So why does it happen? I said, since climate change can lead to increase in temperature. You guys know climate change leads to increase in temperature, and the equator is where the tropics are. So the tropics will be getting hotter, or the temperature will be very high in comparison to the northern part. So the northern part, which was previously cooler or colder, is now going to get warmer. So these organisms, in order to survive, their body physiology works at a temperature that should be appropriate. If the temperature is really very hot closer to the equator, they have to move northwards where the temperature is more friendly or warmer and not that hot enough. So if it's warmer, it means their body physiological processes can occur at the right temperature in order to prevent damage to the enzymes or damage to the organs within their bodies, basically. So I say that species move north away from the equator after climate change. And since climate change can lead to increase in temperature, the temperatures around the equator will be too high, while those towards the north will not be as cold, meaning they'll be warmer. So the species move towards cooler temperatures in order for their body processes to go on well, enzymatically controlled processes. So since enzymes control many body processes, the enzyme's activity is temperature dependent. Again, you know that. Increase in temperature can increase the rate of enzyme activity until a specific optimum temperature, and then after that, denaturation can occur. So if conditions closer to the equator are so high, or the temperature is so high, enzymes could denature, and when they denature, the body physiological processes could be halted, which could be problematic for the organisms. So let's go to the next part. Part C. The effect of temperature on reproduction in a species of beetle was studied. Groups of male and groups of female beetles were each kept at different temperatures for 5 days. The beetles were then mated with beetles of the opposite sex that had been kept at 30 degrees for 5 days. The mean number of offsprings per breeding pair was then determined. So the graph shows the results of this study. In this study, we can see the vertical part is showing us the mean number of offsprings per breeding pair, and the horizontal axis we can see the temperature that the beetles were kept at. Again, remember, the temperature was varied, but the opposite sex was kept at a specific temperature, which was 30 degrees for 5 days. And here we can see X is showing us the females while this is showing us the males. So I will go to the first question. It says, identify 2 conclusions that can be drawn from this study. So from this study, we can see that actually increase in temperature affects males more than it affects females, since after a certain temperature, you can see there is going to be a very low mean number of offsprings per breeding pair in the males in comparison to the females. So I said overall, change in temperature at which the beetles are kept affects males more than females. And then the next part, we can look at the optimum temperature. For the males, their optimum temperature is about 35 degrees since they have the highest. You can see that circle there. That is the highest mean number of offsprings per breeding pair. And for the females, this is the value here, which is about 38 degrees. That is, so the optimum temperature for them. So I say the optimum temperature to keep beetles at before being mated with a beetle of the opposite sex, kept at 30 degrees, is 35 for males and 38 for females. These are the 2 conclusions I can draw from the table or from the results in the graph. So let's go to the next part. So this is the final part of question 4. It says, explain the differences in the results obtained for the male and the female beetles. Every time they tell you to explain something, it means there is a specific fact they want you to state and then give a reason as to why that fact is the way it is. So this fact is why the difference in the results obtained between the males and females. Why is it that females can withstand a higher temperature in comparison to the males? This all lies in the sperm. So I say, based on the results, more successful fertilization occurred in females than in males as the temperature increased. This is your fact. Now you have to tell the examiner the reason as to why you think that is. So I say, this is because the sperms are affected by higher temperature, but eggs are not affected. So the higher temperature damages the sperm, and when it does, it affects its motility as well as viability. Motility is about moving, and then viability is about basically being alive and the capability for it to fertilize a female. Then I said, since sperms have enzymes, these are acrosomes, they're found at the top part in the headpiece of the sperm. The increase in temperature is going to affect these enzymes, meaning the reactions these enzymes catalyze or participate in are not going to be well done, since most of these enzymes could be denatured. So fertilization is going to be inhibited by that. This brings us to the end of question 4, and this is the last question of this video. This is part 1 of the 2 parts video. So please do not forget to subscribe, and I will put a link to part 2 of this video in the description box. Please do not forget to check it out. See you in the next video. Bye-bye.